Imagine trying to fold a paper airplane in the middle of a windstorm while classmates keep bumping into you. It's hard to focus, and the airplane ends up a mess. That's what protein folding is like inside a busy, crowded cell. Now imagine someone steps in to help, holding the paper, guiding your hands, making sure the folds go right. That's what chaperone proteins do. They don't fold the protein for you, but they create the right conditions so folding can happen properly. But what exactly is protein folding? Let's do a quick recap. By the end of this lesson, students should be able to define chaperone proteins and their significance in proper folding and prevention of aggregation and misfolding. Identify key chaperone families and their relevance. Explain the chaperone functions clearly. Link the role of chaperones in clinical diagnostics and diseases. So, quick recap. What is protein folding? It is a process by which polypeptide chain folds to become a biologically active protein in its native 3D structure. These are held together by different molecular forces, and its amino acid sequence determines how it folds. Just because a protein is made doesn't mean that it's perfect. After being synthesized, it has to fold into the right shape, kind of like how a piece of paper can be folded into a paper airplane. It needs to fold in a special way to do its job. If it's wonky, it won't be able to fly far. It's the same way for proteins. If it folds the wrong way, it might stick to other proteins, get broken down, or end up in the wrong place in the cell. The importance of proper folding all comes down to one powerful idea. A protein's shape determines its function. But why exactly is this so important? Well, the specific three-dimensional structure of a protein, known as its final conformation, is what allows it to interact precisely with other molecules, whether that's speeding up chemical reaction, sending signals, or providing structural support. So what exactly happens when a protein isn't folded properly? Sometimes, proteins can lose their proper shape when our cells are under stress, like when it's too hot, there are harmful chemicals, or when the cell is damaged. When proteins fold the wrong way, they can stick together and form clumps inside or outside the cell. These clumps are called plaques or tangles. Let's watch a video to help you visualize this concept.
It takes several minutes. Misfolded or aberrant proteins are not only a waste of energy for the cell, but they are also dangerous. These aberrant proteins typically have exposed hydrophobic regions, which cause them to clump together into aggregates, which can accumulate and cause severe diseases. The cell has two lines of defense against the formation of these dangerous misfolded proteins: molecular chaperones and the proteasome. Molecular chaperones help proteins fold correctly, while the proteasome destroys misfolded proteins through proteolysis. The molecular chaperones and proteasomes compete over these proteins. The longer a protein remains misfolded, either because chaperones haven't reached it or because they haven't succeeded in fixing it, the more likely it is that a proteasome will degrade it.
Alzheimer's disease is a brain disease that happens when certain proteins in the brain fold the wrong way. These misfolded proteins, called beta amyloid, stick together and form thick clumps. These clumps then build up in the brain and make it hard for the brain cells to work and talk to each other, which can lead to memory loss and other problems as people get older. Checkpoint time! Which disease can result from misfolded proteins? A. Alzheimer's disease. B. Parkinson's disease. C. Huntington's disease. Or D. All of the above. Pause the video to think. The answer is D. All of the above. Next question. Based on the video, what process do the proteasomes go through to destroy misfolded proteins? A. Degradation. B. Proteolysis. C. Substitution. Or D. None of the above. The answer is B. Proteolysis. So, we can all now agree that proper protein folding is very important, right? But how exactly does the body ensure that proteins are folded into their right configurations? Well, the answer is presented right here. It is the chaperone proteins. Chaperones are proteins that aid in the proper folding of other proteins by facilitating their assembly without being a part of the resulting complex. So they act like catalysts, helping the folding process happen faster but don't give out instructions. Some proteins are like tricky puzzles. They're hard to fold because of their complicated shapes. If they fold in the wrong way, they won't work. Think of a protein like a key. It has to fit just right to do its job. Chaperone proteins don't change the pieces. They just help everything fit together so the protein works the way it should. Another checkpoint. What is the correct definition of a chaperone protein? A. It is a human cell. B. It is a part of the final result. C. It allows aggregation during the process. Or D. It prevents misfolding. The answer is D. It prevents misfolding. There are two main types of protein helpers, molecular chaperones and chaperonins. These differ on how they assist protein folding. Let's start with molecular chaperones. These proteins attach to unfolded or partly folded proteins to keep them stable and prevent them from clumping or being destroyed. A key example is HSP70 or heat shock proteins, which bind to sticky parts of a protein and uses energy from ATP to grab and release it in a controlled cycle. It even keeps some proteins unfolded when they need to move across membranes. HSP70 does not work alone. It gets help from co-chaperones like HSP40 in humans or DNAJ in bacteria, which make the process faster and more efficient. Later on, I'll be showing you a video to help you visualize this mechanism. Another molecular chaperone is HSP90, which helps special client proteins become fully active and functional. It also works in an ATP-powered cycle, functions in pairs, and plays a role in removing proteins that are too damaged to be fixed. Chaperonins, also known as HSP60, are special helper proteins that act like tiny folding rooms. When a protein is unfolded and needs help, it enters this barrel-shaped chamber to fold safely without sticking to other proteins. There are two main types of chaperonins, Group 1 and Group 2. Group 1 is found in bacteria, mitochondria, and chloroplasts, and uses a separate lid to close the chamber. GRU-L and GRU-S is an example, where GRU-S is the lid and GRU-L is the chamber. Group 2, on the other hand, is found in the cytosol of eukaryotic cells and in archaea. Its lid is built into the chamber itself. Both types uses energy from ATP to open and close the folding chamber and help the protein fold the right way. Next checkpoint. Which major chaperone protein family works with co-chaperones to complete its process? A. HSP90, B. HSP70, C. HSP100, or D. HSP60. The answer is B. HSP70. Now, we'll watch a video to help you better visualize the two main families and the mechanisms of HSP60 and 70 of chaperone proteins.
The more likely it is that a proteasome will degrade it. Let's discuss chaperones first. Chaperones work by binding to exposed hydrophobic patches on misfolded or incompletely folded proteins and hydrolyzing ATP. Some of the energy expended by chaperones is used to perform mechanical work, but much more is used to ensure the accuracy of protein folding. The proteins that chaperones help fold properly are called client proteins. There are several families of molecular chaperones in eukaryotes, which function in different organelles. The ones we will be discussing are HSP60 and HSP70, each of which has a set of associated proteins. HSP stands for heat shock proteins because lots of them are produced when a cell is exposed to elevated temperatures, responding to increased amounts of misfolded proteins. HSP70 acts early on in the process of protein folding, binding to polypeptide chains emerging from ribosomes where there is a chain of 7 hydrophobic amino acids. HSP70 is aided by HSP40s, and many cycles of ATP hydrolysis are generally required to fold a single polypeptide chain correctly. HSP60 acts on fully synthesized proteins, forming a large barrel that serves to isolate the protein and provide better conditions for folding. HSP60, which is a type of molecular chaperone called a chaperonin, initially captures proteins along the rim of one of its barrels. Then, the binding of ATP and a protein cap increases the diameter of the barrel and partly unfolds the protein. Inside the barrel, the protein has a chance to fold in a more favorable environment. 15 seconds after ATP binds to the HSP60, it hydrolyzes, weakening the complex. The binding of another ATP molecule makes the protein leave. If the protein is still not folded correctly, it can repeat the cycle. Proteasomes are so important that they constitute almost 1% of cell proteins. They are dispersed throughout the nucleus and cytosol. If a protein folds correctly and quickly, then none, or only a small bit of it, gets degraded. A slowly folding protein is at risk for a longer time, and more of it might get destroyed before the remaining amino acids attain the correctly folded state.
I hope this video clarified what I've discussed earlier. Now, I will ask you another question. What happens when a protein is still not folding right, even with the assistance of chaperones? Well, if you can remember in that video I showed you earlier, proteasomes were mentioned. Proteasomes are a large protein complex that breaks down unwanted or damaged proteins. These require metabolic energy to function and works closely with ubiquitin. When scientists were studying how chaperones chose to fold or destroy proteins, they found out that co-chaperones play a key role in this decision. One of these helpers is BAG1, which works with HSP70. BAG1 basically tells HSP70 when to let go of a damaged or misfolded protein. Once released, if the protein is still not working right, it gets tagged with a tiny Destroy Me label called ubiquitin and sent to the cell's recycling center, the proteasome. This is called the ubiquitin proteasome system. Now for our last checkpoint, which of the following breaks down unwanted and/or damaged proteins? A. Water, B. Proteasomes, C. Chaperonins, or D. None of the above. The answer is B. Proteasomes. Since this discussion is somewhat introductory, if you want an even more detailed discussion on the different types of chaperones, I highly recommend watching this video or watching the full video. Here are our references as well. Just pause the video, and if you want a recap on the topic, or if you want a way to evaluate yourself, we actually made a video of us playing Jeopardy with the topic of chaperone proteins. Go check it out if you want to quiz yourself or just follow along with the game. Here are some short clips of that.
So look for 2K points to win the prize. So the prize would be player 1 would be Miss July, and player 2 will be Miss Ysabella. So let's start, everyone.
Okay.
So
As Linus Pauling once said, proteins hold the key to the whole subject of the molecular basis of biological reactions, reminding us that proteins and their proper folding are essential for nearly everything a cell does. So, if the tiniest misfold can cause such big problems, how much power do we hold by simply understanding the fold? Well, future medtechs, I hope you learned something. And in case I don't see ya, good afternoon, good evening, and good night.